Well, thanks for joining us. What I'm going to do is, as most people know who are following NIH activities, this is the year we renew the networks. So the four networks that are going to go forward are the Vaccine Network, the Prevention Trials Network, the Impact Network, which deals with moms and babies, and the AIDS Clinical Trials Group Network. All four of those networks are going to be renewed with the leadership that I think you're familiar with. What I'm going to try and do is talk really fast, slowing down only when I need to, about how we're thinking about the way forward for the Prevention Trials Network, which gets into a lot of technical detail about the tools and the strategies. And then I hope there'll be time for questions at the end, and then people can write me if they have questions beyond that. So the first thing I want to talk about is the U.S. has had a kind of sea change in its activity, and the Health and Human Services in the U.S. has decided that the epidemic in the U.S. is so constrained that it would be possible to use the existing strategies, most of which were developed by the people on this call over time, the existing strategies to end the epidemic in the United States. And these are a couple of articles citing that plan. And the reason why this is believed to be possible, because the U.S. epidemic is so focused on African American men in the South, as shown on this slide. So most of the interventions that will go forward using the tools that we'll discuss will focus on that particular group, geography as well as race and gender. And in fact, this is divided in the United States into counties, and certain counties are going to receive a lot more activities than other counties. There's like 5,300 counties in the United States, and less than half will get any activity, and even a smaller proportion will get the majority of activities. And the pillars of the intervention are increased testing and diagnosis, improve the treatment cascade, use PrEP for people at the greatest risk, which in this case is going to mostly be African American men at risk, and then break up clusters using social network theory and phylogeny where possible, although phylogeny is ironically ethically challenged in the United States. There's a lot of concern in the risk communities about using this tool. But these are the kind of the four pillar strategies. These four pillar strategies kind of evolved from work that's been going on for a very long time in the Prevention Trials Network and beyond. On this slide is everything we know how to do. We'd like to make a vaccine. We don't have one. We have other tools that would be used for pre-exposure prophylaxis. We'd like people to have safer sex behavior. Of course, that's proved to be not sufficiently effective for our purposes. And then ultimately, we've used treatment as prevention, testing and treatment as prevention as a key strategy. And I think everyone on this call is familiar with the idea that in the end of the day, HIV, sexual transmission of HIV is kind of a one-off situation generally with one person infecting another person. That can be a straight couple or a gay couple. And everyone in a relationship is either in a concordant negative relationship, which is most people on the planet, a concordant positive relationship, which ironically was believed at the time we started this work to be dominant, and I'll get back to that in a second, or a discordant relationship. When we started investigating ways to evaluate treatment as prevention, it was generally believed by both patients and providers that if you're infected, your partner must be infected. But in fact, most studies show less than half the partners infected at the time an index case is discovered. So the focus on partner prevention is not trivial. And basically, as I think the people on this call know, that in 2011, the 052 study and the surrounding work was identified as the breakthrough of the year, as it demonstrated virtually complete prevention of HIV when viral suppression had occurred. This is my most expensive slide, by the way, because the guy's mouth, Bruce Alberts, then the editor of Science, his mouth moves in this slide. So that took a lot of effort to make his mouth move. All this work led to the 90-90-90 campaign, more recently the U=U campaign, and it led to focus on the continuum. Before all this, we didn't focus so much on linking people to care and getting viral suppression, but this is now the sine qua non of all prevention strategies, coverage and linkage to care. And it led to the four large trials that I think everyone on the call is familiar with, where I think these trials demonstrated unequivocally that treatment serves as prevention, but imperfectly. And I think the PANGEA group has come up with the questions that are on the slide. What's imperfect about treatment as prevention? The magnitude of coverage, the possibility of resistance in some population, and then untreated people in specific populations, including people with acute infection, people migrating into communities, young men in the United States, young black men as a key population. And I think what's important is if we're going to better refine treatment as prevention, we need the attributable risk of each population. And I know the PANGEA group is working on defining the attributable risk of each population using phylogeny and a lot of other theoretical activity. Now, so on the infected person side, we have treatment as prevention, and that's really kind of considered axiomatic. And I know you all know last year, first it was demonstrated for heterosexuals, and I don't want to forget the idea that the Alison Roger and her group and many other observational studies demonstrated that the treatment as prevention strategy works for gay couples as well as straight couples, or a better way of putting it is isn't affected by which sexual behavior is encountered. So anal intercourse treatment as prevention prevents both HIV acquisition from anal intercourse and heterosexual intercourse. So the flip side of this coin is going to be strategies for prevention for the HIV negative person, the ultimate strategy, which we'll get to at the end of all this, being HIV vaccines. As everyone on the call knows, TDF-FTC, Truvada, was approved by the FDA many years ago, and it's proved to be a remarkable home run out of the gate. That is, the combination is very effective for preventing HIV acquisition in men and women. But the real, and PrEP use has increased, as shown on this slide, which was presented a couple of years ago. Now there's probably about 300,000 to 400,000 people worldwide using PrEP. But in reality, more than half the people using PrEP are living in the United States. And in that group living in the United States, we still have a male distribution of the wrong people getting PrEP. So most of the PrEP delivered, there may be a slide about this in a minute, most of the PrEP is being delivered to gay white men, whereas what we really need is for it to be delivered in the United States, at least to gay black men. And then, of course, we also need it delivered all over the world to the highest risk groups, and that has not transpired. The other problem we have with PrEP is shown on the next slide, that we start these pills, and we give people these pills, and there's a very brief window where adherence is excellent, and then adherence or PrEP continuation, or persistence as it's now known in this field, falls off dramatically. And whether it's in men in the United States or young women in sub-Saharan Africa, as will be published soon in a study called HPTN 082, whichever population you look at, it's hard to find more than half the people taking PrEP at 6 months. And in many groups, it's much lower, as shown on the slide. After a year, maybe 20% of the people who started PrEP are still taking it. So the discontinuation rate is very high. And lots is going on to try and understand, can we improve adherence with these pills? Can we better understand the discontinuation? And we do recognize that at the time of discontinuation, while we think, people might be discontinuing their PrEP because they're no longer at risk, we see substantial number of seroconversions in people at the time they discontinue PrEP or sometime after they discontinue PrEP. So I think it's unreasonable to believe that there's a sensible discontinuation strategy going on. We're right now working on a paper about this with WHO and the CDC, which I think will be published as a review in Lancet HIV. So that's oral daily Truvada PrEP. It's probably worth noting that oral daily treatment is preferred by some people, not by others. It's probably fairly essential for women at this point, but men can benefit from a 2-dose, 2-1-1 strategy. The WHO has endorsed that strategy. CDC has yet to endorse that strategy. But I believe that 2-1-1 for men will become more popular in the coming years. Now, I think everyone's aware that there's a controversy surrounding a newer combination of the alphinamide version of tenofovir TAF plus FTC compared to TDF-FTC. And a study was done called Discover, using this combination Descovy. And I just want to say a few words about this. So this combination was chosen for a variety of reasons, and it proved to be effective in a particular way. But the rationale that Descovy is better than Truvada, there are many investigators who believe that there's not a lot of evidence that one is better than the other. The tenofovir agent probably does cause more loss of bone density, has some problems with mild change in renal function. That is not observed with the Descovy combination, the TAF-FTC combination, but the cost-benefit ratio may not line up strongly in favor of Descovy. Nevertheless, we still anticipate, for a variety of marketing and other reasons, that Descovy will probably replace Truvada in the coming years, especially as it gets cheaper. Now, the only thing I want to point out on this slide is when this study was set up, it was set up to have like 150, this was done in gay white men in the United States and Europe, it was set up to have 150 incident events. But it turned out that the people in this study took their drugs very reliably, and because they took their drugs reliably, there was very little incidence. And fortunately for the investigators doing the study, the way the ultimate incidence lined up was that people were more reliable with their Descovy than they were with their Truvada, and so Descovy efficacy was slightly better than the Truvada efficacy, but both were very efficacious. The only thing this raises is the issue that going forward doing clinical trials, if people are going to be adherent to whatever intervention we're using, it's very difficult to do non-inferiority superiority studies because everyone's going to benefit. So the FDA has recently indicated to the group that does Descovy research that they can use other strategies in clinical trials to prove what might have happened. And the strategy that is being embraced at some level is a counterfactual strategy. We don't have time to talk about all the ins and outs of this today, but I think that's worth talking about at a future moment in time. The power and the weaknesses of counterfactual strategies. The other problem with this study, the Descovy study, while the people were adherent to the pills, the communities that were being investigated also had a lot of treatment. And of course, if people are treated, they're no longer contagious. So the treatment side will compromise interpretation of PrEP efficacy, whatever PrEP we're using. And so we're in kind of a new window of consideration about PrEP. Now, an alternative universe is longer acting or alternative ways of delivering prevention. And what attracted the greatest attention over the last, I'd say, 10 years was the idea of using a topical agent in a ring or topical agent in a gel, but preferably in a ring because it could be distributed over a longer period of time. And the topical agent that was tested was Dapivirine in a ring. And the good news about the study is that some benefit was observed, maybe 50% benefit across the study, mostly in older women. And kind of the bad news of the study, that only 50% benefit was observed. And Dapivirine is not a drug that's metabolized. So if the ring is removed at any time before or shortly after intercourse, then the benefits of Dapivirine will be lost. The other problem is that that these drugs, topical agents, will only diffuse so far. And so I think it's not reasonable to anticipate that any topical agent, or very few topical agents, would be comparable to a systemic agent. Now, the Dapivirine rings went to the EMA, the European agency, like FDA, for approval, and they've been waiting for more than a year to determine whether there'll be approval or no approval as they stand today. I heard that the approval decision will be made in May. I think the most important thing for the networks or the PTN going forward is the possibility of having other drugs in a ring, if we were going to do it, a drug that would be metabolized, but probably more importantly, multi-purpose technologies where you would try and prevent pregnancy and HIV concomitantly, where maybe imperfect HIV benefit would be acceptable, or STDs and HIV concomitantly. This multi-purpose technology issue is a big deal to us. The alternative that's going on right now is the generation of a pill of Truvada plus oral contraceptive, a daily pill. So you can see for young women who are willing to take oral contraceptives, that kind of combination, which is being generated by the population console, the pop console, that might be an attractive alternative. So in summary, rings are a way to deliver a topical agent for a long period of time, and we'll wait and see what they do with the piperine. There are alternative rings with other agents being developed, and the rings have potential for multi-purpose technology. So most of the energy in this field, the tool development part of the field, has gone into longer acting agents. This slide, okay. And the first of the agents that's come up for testing is cabotegravir. The PTNs worked very hard in collaboration with Gilead, with Aviv, GSK and Viv, and with the Gates Foundation in trying to bring this drug to the marketplace for prevention. Gilead's a partner as well because our studies here are going to be in comparison of cabotegravir to Truvada, which is a complicated comparison. Now, cabotegravir is a drug that's 97% the same as dolutegravir. The difference is it's been milled into a nanosuspension, and the nanosuspension goes into the muscle and subcutaneous fat, and there it's slowly released. So the drug doesn't have a long half-life. It's the deposition into the tissue that allows infrequent dosing. And so dosing as infrequent as every 2 months or 3 months, but our studies have been 2 months, is sufficient to achieve levels that would be predicted to be protective. Now, the good news is that that's great. You give a shot and no one has to come back for a couple of months. The bad news is we can't get the drug out, and the drug is going to be around for a very long period of time. In this study and a subsequent study that I might have been published by now, I can't remember, we show that the drug is going to be around for a year, not at therapeutic levels. There's going to be a therapeutic range, a range that might force resistance, and then a subtherapeutic range that is inconsequential, but for a very long period of time. And women will have a much longer half-life than men. Women, because of fat and muscle organization, the amount of drug detectable in women will be more than a year. So the good news is you inject the stuff and you get prevention for a very long period of time. The bad news is we can't get the stuff out if we want to get the stuff out. Having said all this, we've gone on to set up clinical trials, and there's kind of a parallel universe. The company itself put together rilpivirine injectable plus cabotegravir injectable and showed, this is Viv and Janssen, showed that combination, after a run-in with some pills, is comparable to triple drug therapy. They're waiting for FDA approval. They sent a letter to FDA seeking approval in December. FDA put them on hold for a short window of time because they needed more information about the manufacturing of the product, not because there's any evidence of safety or efficacy concern of the product. So we've gone on to do 2 parallel studies. One's called HPTN-083, the other's called HPTN-084. HPTN-083 is a study in men in the Americas. HPTN-084 is a study of women in sub-Saharan Africa. And by the way, 083 is in the Americas and Asia. Basically, there's an oral pill run-in, and the only reason for the run-in is in case somebody has a side effect, we don't want to inject them. So far, there have been no serious side effects during the run-in. And then there's this study of the injection blindly compared to Truvada. So it's double-blind, double-dummy. So everybody gets an injection, and the injection is either going to be placebo or the active cabo product, and everybody's going to get a pill, and that's going to be a placebo or active Truvada. And that study goes on for some long period of time. People are receiving these injections over several years, and then when the injection phase ends, which is capped at 3 years at this point, when the injection phase ends, and we may not need people to go 3 years, but that's to protect the integrity of the efficacy of the study. At the end of that study, because there's that tail of cabotegravir, that everyone in the study gets Truvada for a year following the injection phase of the study. And the 083 study is being done by Rafi Landovitz and Beatrice Grinstein. It's got enrollment, it's fully enrolled with 4,500 men at 27 U.S. sites, 11 South American sites, and so on and so forth. We are, for a variety of reasons, adding 500 more subjects in South America and Asia. The women's study is got almost 3,000 women enrolled. It's just about fully enrolled. It's conducted at the sites shown on this slide in sub-Saharan Africa. It's being led by Mina Hassinapur and Sinead Delaney. Both studies are really, going well, and hopefully will prove that cabotegravir is either not inferior or superior to Truvada. Now, there are challenges with studies this big. There's recruitment and retention. Recruitment has been excellent, but retention to injections is going to be challenged. And so some modest percentage of people are going to drop out of the study. That compromises interpretation. If everyone takes so this study is also predicated on the idea, based on slides I showed you earlier, that people are going to stop taking their Truvada. But if everyone takes all their pills, then incidence is going to be very low, and we'll run into the same trouble that the Discover trial ran into, of how to interpret very low incidence in both groups, the experimental group and the control group. Another issue that we have not yet talked about are inflammatory sexually transmitted diseases. The miraculous thing about Truvada PrEP is, even if you get an STD, but you're taking Truvada PrEP, and you get a lot of inflammation or an ulcer from your STD, Truvada PrEP still works. Because STDs are very common in people taking PrEP, we need to prove that cabotegravir, LA, long-acting PrEP, will also not be overwhelmed by STDs. So we're carefully monitoring STDs. And lastly, without going into this in too much detail, it is complicated as we get to the efficacy of these studies, it's going to be complicated to look at the intention to treat analysis versus the as treated or on drug analysis. If there's time, I can explain this in more detail, but it is going to be challenging for these interpretations to transpire. We have every 6 months, we're totally blinded to all results. Every 6 months, we meet with our Data Safety Monitoring Board. The next meeting for these studies is in May. Every study meeting is a drama, because at any time there may be sufficient benefit in the cabotegravir arm or sufficient failure to stop the study. So in May of this year, we'll get our next peak, not a peak of results, we'll get information about whether we're going to go forward for another 6 months or whether either one of these two studies will be stopped. Now, I've talked a lot then about cabotegravir and tenofovir and FTC, but there are other drugs in development, and the one that's attracted the greatest attention is Merck 8591, now called islatravir. And islatravir, shown on the slide from monkeys, is if you give islatravir orally, this NNRTI, you get complete protection from exposure to HIV in a rhesus macaque. This generated a lot of excitement because this drug is very, very potent. So a single pill, because of its potency, can last a month. It works by a particular magical mechanism that's beneficial, very long half-life, and once a month dosing for prevention seems possible with this oral pill. And this is just an example of a clinical trial that Merck has set up in clinicaltrials.gov of this once-a-month idea that I've expressed already. But I think the excitement beyond once-a-month pill is in an implant. That brings us to consideration of another way of delivering drugs for much longer periods of time. Of course, implant depot contraception that lasts for 3 months, or implant contraception, is very popular in some parts of the world. And Merck, who make the main birth control implant, right off the bat said, wait, we can make a MK8591 implant, and they've already succeeded in making the implant. They know how to put the implant in because it's the same exact device for which they use birth control. They've already tested this in humans and demonstrated that they can get very substantial, very long release of MK8591 through their implant. And that kind of brings us to the implant competition. So for the implant competition, you've got two kinds of implants. a matrix implant, which is like the 8591 implant, where the drug, the MK85, it's integrated into the implant material and it's leaking out of the implant material. The alternative is a reservoir, like a big chamber where all this stuff is there, and there's a little hole in the chamber or holes in the chamber that let the stuff leak out. The problem with a reservoir is if dumping. You can't put so much drug in the implant that it might all be dumped at once, or if it is dumped at once, you got to be sure it's safe to dump it. So those are the two kinds of strategies, and then there are some implants that can dissolve. They're biodegradable, and others are renewable. They can be, you can shove more drug in, but the renewable ones would have to be removed eventually as you remove the current Merck birth control implants. The drugs competing for implants are 8591 is Islatravir, Dapivirine, which has an implant that's in investigation supported by NIH, being done by Northwestern. Several investigative groups are doing TAF implants, Oak Houston Research Triangle Institute of Northwestern. And then there is a Dolutegravir idea that I think I have a slide about called the cell gel idea. Cell gel idea is like this. This is being done by my colleague Victor Garcia with the collaboration of Martina Kovarova. And what they've done is they've been able to take drugs, but in particular MK8591 is Islatravir, and they can inject it as a liquid into the soft tissue of an animal, and then it becomes a solid, and the solid is a lump, and the lump releases drug for as many months as you want. Depending on the size of the lump, you get drug for 3 months, 6 months, a year, and you can take the lump out. If you had to, you could surgically remove the lump, although in the end of the day, one would hope to have a lump that would eventually disappear. So the cell gel idea has been supported by the Gates Foundation, who recently provided funds to further pursue cell gel. And this is not dissimilar to work done at MIT, where they try and put drugs into the stomach, and they don't get past the stomach. They don't get past the stomach into the duodenum, and they sit in the stomach for a long time and deliver drug. And this is just an example of Victor's work with humanized mice, showing that the Islatravir in humanized mice delivered as a sol gel can get you complete protection of HIV, or almost complete protection, good protection, relative to a placebo. Okay, now the final thing I want to talk about before we get to questions has to do, so we've got the treatment side and we've got the prevention side, and we've got the antiretroviral drugs we'll think of as small molecules. But then there's the alternative, which is a hugely important alternative, is let's make a vaccine, because most people believe you cannot really end the AIDS epidemic without a vaccine. My colleague Christoph and others on the call are mathematical modelers, and they've done a brilliant job at showing how we can put a lot of things together and get benefit. But I think a highly efficacious vaccine is the holy grail in this field, a cure and a highly efficacious vaccine. So the vaccines right now that are being developed are in two flavors. One flavor is kind of the traditional non-neutralizing vaccine that somehow might inhibit the growth of the virus or invoke a antibody-mediated cytotoxicity response that limits replication of the virus, either so that a person can't get infected, or if they do get infected, they have a lower viral load and they can't infect the next person. And currently, what drove this field in the non-neutralizing or the now called the non-bnAb antibodies, what drove the field was the RV-144 study done by the Army, where they took stuff that evoked antibodies against envelopes, and they saw a modest benefit, a 44% benefit over a single year, and that led to a huge set of experiments to try and understand why the benefit was observed. Some biological plausibility surfaced, and then somewhat similar vaccines then were developed. One vaccine, the kind of parallel study to the RV-144 study, trying to get more than 44%, is called the P5 collaboration, or HPTN 702. And then Dan Baruch and Betty Korber and others looked at all the envelopes that they could find and all the kind of neutralization, in vitro neutralization strategies they could envision, and they made what's called a mosaic vaccine that's being tested by Crisell and Janssen and the Gates Foundation, and that both those studies are kind of in the field. So that's kind of one approach. I would call it a traditional approach of a vaccine, of repetitive doses of the same vaccine to invoke sufficient immunity to prevent infection. But there is another idea, and the other idea is that there are broad neutralizing antibodies, and that they could be used as passive immunity, and that by using them as passive immunity successfully, they can help drive the generation of a vaccine that makes an antibody or some antibodies that are similar to broad neutralizing antibodies. So we need to talk for a few minutes about broad neutralizing antibodies. And I apologize that this is a lot of different topics in one lecture, but this is what Lucy commanded me to do. So here's how this works. The Oxford group played a big role in Oxford and Duke and other investigators played a big role in our understanding of this. But basically, when HIV is acquired, an immune response occurs very quickly, in a few days and then early weeks, and that immune response is antibody-directed in envelopes, and there's a T-cell response as well. And the antibodies are the antibodies we use to measure to prove someone's infected. Those are the diagnostic antibodies we use. And those antibodies do not prevent the replication of the virus. And the reason they fail to prevent the replication of the virus is as HIV replicates, it lacks fidelity. And so very quickly, mutants are generated that escape the antibody that was generated. But what happens is that the host does not give up. The host has literally billions of B cells available to try and make a successful antibody. So the B cells of the host go on and they make another antibody. And that other antibody then says, I'm ready to neutralize HIV, but by the time it gets there, the HIV escapes. This arms race, or this war between the virus and the host, goes on and on and on. And it was recognized many years ago that some percentage of hosts in this war make antibodies that will not neutralize their own virus, no matter what, and don't contribute to their own health, but that they're very broad. And so those are referred to as broad neutralizing antibodies, at least in an in vitro test that was developed by John Miskell and David Montefiore, in an in vitro test that's divided into tiers of resistance. The broad neutralizing antibodies will kill a large percentages of all the strains that they put into this in vitro test. 80% or 90% or 95% of all the viruses put into the test. So the bnAbs, and at first there was just a very small number of bnAbs, and now there are hundreds of bnAbs, because you can find bnAbs in 10% of people with HIV as long as they're untreated. If you treat people, of course, you end the arms race. So a lot of the bnAbs are generated from people who are no longer alive, where we have their cells, and through technologies that were developed only about 10 years ago, you can isolate a single cell and find a bnAb. And bnAbs then have like 3 features that will make them attractive. First of all, can a cell line make the bnAb with efficiency? Because you need to be able to make the bnAb in sufficient concentration to infuse or learn something about it. And some bnAbs are very difficult to make in vitro. Second thing is potency. How much antibody does it take to kill the virus? The more potent bnAb is more popular. And then breadth. How many viruses will it kill? 50%, 60%, 70%, 80%. So that's the kind of bnAb ontogeny. And the bnAbs don't all work at the same site. This is just a map of the HIV surface, and you see that many of the more popular bnAbs attack the CD4 binding site, where the virus has to bind to the CD4 site of the cell. Some attack the VC3 glycan, some attack the mPER site, and others attack the V2 glycan apex site. And these 3-dimensional crystallized structures are reviewed with every bnAb. The bnAbs, on this slide I'll tell you, the bnAbs that attract the greatest attention are the VRC series, 01 and on, the BNC Rockefeller antibodies, and there's a few of those, the 10-10-7-4 antibody, and the CAP256 antibody. Each are different. The 10E8 antibody is actually the best antibody, but in recent studies proved to be toxic. And so it's kind of on hold right now. And if you took VRC01 and you put it into an animal, and you give the animal a vaginal challenge or a rectal challenge, the VRC01, and you've chosen a virus that's susceptible to VRC01, and you can prevent infection. And VRC01 has modest potency, pretty good breath, 80%, 90% breath, so it's a reasonable antibody, but it's a first-generation antibody. Now, then, with this first-generation antibody available, made by John Mascola at the NIH, a decision had to be made about 4 years ago, roughly, maybe almost 5 years ago now. The decision was, we know there's better antibodies in development. Are we going to wait to see what the better antibodies are, or are we going to start a trial with what we have? Are we going to love the one that's available? So a pretty big decision was made to go forward and test VRC01. And that decision was not based on the belief that VRC01 would be a product for prevention. It was to determine whether it was safe and how you would do safety studies, whether efficacy studies were possible, how you would ethically do efficacy studies, and probably equally important, how to design all the statistical and biological frameworks necessary to see if a bnAb works. And I'll get into that in a little more detail in a second. So a decision was made to go forward. And so Larry Corey and I are the leaders of studies called the Antibody-Mediated Prevention Studies, and Niradza Magodi and Surya Jabaganti are the protocol chairs, or the protocol team investigators. One of the studies is called HPTN 085, HPTN 704 in men who have sex with men in the Americas and Europe, Europe being Switzerland, and the other studies in women in sub-Saharan Africa called HPTN 081. And these studies are fully enrolled with, I think, about 7,000 subjects. And the studies had 90% retention, incredible retention. And the studies are going the full distance. That is, they weren't stopped for futility, nor were they stopped for efficacy. And so we are unblinding these results in August, and we're presenting the results at R4P in October. And it's going to be a complicated set of presentations at R4P where we describe how you do these studies and what we learned about VRC01. And the VRC01 studies are placebo-controlled, where you get 30 milligrams per kilogram every 8 weeks, or you get 10 milligrams per kilogram every 8 weeks, or you get a placebo. Now, you would say, wait, how can you give a placebo? What about prevention activities? So everyone in the study gets a prevention package, and the prevention package can include Truvada PrEP. So those people who want to access Truvada PrEP for most of the countries, we're able to do that. They get it for free, and then we monitor drug in blood in all the subjects through a statistical package. So even though we're especially concerned about people on PrEP by prescription, so you can imagine how complicated this is, because we need to understand, is it the VRC01 that's preventing HIV infection, or is it the Truvada PrEP that's preventing HIV infection? And some modest percentage of people choose to go on Truvada in the study, and some modest percent of people continue the Truvada in the study. How do I know the numbers? Because we're unblinded to who gets the PrEP. We're unblinded to drug in blood. By blood spots, we see who's taking the drug. So as I've indicated, the AMP trials will come to an end, and the result will be presented in October, I guess, whenever R4P is. Now, this complicated slide shows you how this really works. So I'm going to go slowly over this. In reality, you've given somebody the highest dose, 30 milligrams per kilogram of VRC01. We know that's going to achieve a concentration of more than 100 micrograms per milliliter of drug in blood at the time of infusion. So the real question is the 100 micrograms per mil of drug sufficient to do, and it's going to be around at 100 micrograms per mil for about 3 to 4 weeks. It's going to wane, of course, but it's going to be a high concentration for about 4 weeks. So the real issue is people who are exposed in the active arm in the first 4 weeks of the study. Number one, do they not get HIV? Do we avert HIV infections? And will we really have the power to see that? An equally important question is, for people who actually get HIV in the first 4 weeks after infusion of the highest dose of VRC01, will we only see variants that are resistant to VRC01? You see, efficacy in the study is not just avoiding infections, it's proving that we averted sensitive infections. Because if we see sensitive infections, it either means the assay we use to measure sensitivity is wrong, or the drug doesn't work, or that the drug never gets to the right tissue at the right time. So there's a lot of drama. This is a study that costs a lot of money. It's gone on for now 4 years. It's got 7,000 or 8,000 subjects enrolled, thousands of people working on the study at 44 sites around the world. And you can imagine we're very anxious to see these results and present the results, and they will be presented in October. Now, whatever happens with these results, this field will go forward, because these results were designed to inform the future. And so on this slide, you see that the first event is VRC01, and then we have another drug called VRC07523LS, which now lasts 6 months instead of 8 weeks. So now we have a much longer acting version of this. And then there are several other antibodies that have also added an LS mutation that makes the drug last longer, the antibody lasts longer. So we anticipate the next trial will be combinations of 2 or 3 antibodies given concomitantly to prevent infection. The AMP trials were done with IV drugs. The next trial will be done with sub-Q drugs. And so I would stay tuned. In 2021, we'll start the next trials. I can share that I think it's common knowledge now that Gilead bought the Rockefeller antibodies that are called BNC117 or 123, and I think they have access to 10.10.7.4. And so Gilead and the Gates Foundation, you can anticipate, might want to do a trial of those two drugs. I can share that the NIH is most focused on 3 drugs, VRC07, PGT121 or N6, which is owned by ViiV, and PDGM 1400. And so these are the kind of drugs that are the highest priority right now. The extra drug, the additional drug that's getting a lot of attention is called CAP256. That's made by the the Durban KwaZulu-Natal site, Caprisa. And that drug is not very broad. It's got 60-70% of clade C, but it's very potent. Just a smidgen of the drug is sufficient. So CAP256 is also being tested as a drug that might fit into one of these combinations. So you can anticipate sub-Q combinations of 2 or 3 drugs tested in the next 2 or 3 years using the infrastructure developed by AMP. So that's kind of, to leave time for questions, that's kind of where we are with all of our tools. Now, if you look one step further, what we really need to be doing is what are called integrated strategies. And so the Prevention Trials Network is integrating treatment as prevention, PrEP, and then all kinds of other kind of behavioral and structural tools into one comprehensive package. That will be used in the United States, in the southern United States, in a study called HPTN 096, the most comprehensive package we can put together, but it's flexible. So if new tools develop, we can insert a new tool during the course of the study. So in summary, just leave about 10 minutes for questions if there are any. HIV prevention research has lots of results. And those results, it's not like anti-science. Those results that I've kind of showed you have driven the current strategies for prevention and will continue to drive the strategies. I think the contribution of PANGEA is obvious, where we're looking for guideposts of attributable risk and to whom to deliver these strategies for the maximal benefit. TASP and PrEP, accordingly, need to find their ways into integrated strategies as best as we can do them. Global HIV prevention right now remains too unfocused for maximal benefit. What do I mean by that? We can't concomitantly prevent HIV in every risk group. We really need to focus our resources on those people at the greatest risk and in whom will benefit the most from the strategies and tools that are now available. We're trying more and more in these kind of special populations, the risk populations, We're trying to more and more get focused on those populations. We're trying to use risk scores and other devices to better understand to whom we should deliver these devices. And that's where this cluster busting comes in, of using phylogeny and other information to try and break up growing clusters. And the U.S. End the Epidemic campaign, which I've talked about, is a big deal to us because it's going to be our first major attempt at delivering an integrated strategy towards drastic reduction of HIV incidence, trying to get a 90% reduction in incidence over 10 years. So that's kind of where the HIV Prevention Trials Network is. This is just about everything we're doing. I want to thank Lucy for inviting me, and thank you for listening. And I'm glad to answer any questions or comments or arguments about all this stuff. So thanks, Lucy. I'm going to stop.